All right, so good morning, everybody, once again. So we're going to do the final lecture, which is going to basically be a recap lecture of what we've learned. And what we've done is, after this lecture, we're going to be holding a quiz just to kind of, see how well you've understood. It's not meant to be a competition. It's not as if we're going to check your scores and we're not going to do a comparison. It's you versus you. The most important message here is I would like to see if you really had, made some progress in terms of the understanding of the coursework that has been, delivered to you, right? So feel free to, take any notes during this whole recap thing, because most of your quiz is based on what we've learned. There's nothing going to be coming somewhere, under the blue sky kind of questions here. And they're going to be clear multi-choice questions at the end of this particular lecture. Now, this lecture is meant to be a recap lecture because, most of the area that we've covered as part of the whole GAN series have been, topics that are completely new, topics that are completely, that are kind of in initial stages. There's a lot of research work going on, and I've been giving you examples from my own research group. So we're going to carry on the same lines, and we're going to look at, again, a recap lecture here, and we're going to go over the whole thing one more time. All right, so welcome to this final lecture, which is on the recap of all the contents that we covered as part of the GAN series here. So I think one of the things that we learned here is about electrochemical biosensors. We've restricted this whole GAN lecture series on electrochemical biosensing, right? Because electrochemical biosensors, as we all realized, they're more easy to miniaturize, and then if you can miniaturize things, you can easily implement them in the field settings, right? Rather than having your electrochemical sensors, which are based on huge big machines which either sit in a lab or sit in a big hospital, in a specialized hospital. You need something which can be more point of care, right? Therefore, we spoke about electrochemical biosensors. And as part of this electrochemical biosensors, what we saw here is basically you can have different types of samples where you can use your electrochemical biosensors. You can have cell culture samples, you can have human samples, your blood, urine and saliva samples, your food samples, or you can have environmental samples. And then you have your biosensor, which is right in the middle of that, which basically would have your transducers, you would have your bioreceptors, and you would have your electrical interfaces. In bioreceptors, you can have different types, which will be, in bioreceptors, in terms of bioreceptors, we could have different types of bioreceptors. We'll cover them a little bit of more detail in the following slides. And then you have your electrical interfaces, which include things like, you've done an experiment yesterday as well. Can anybody tell me what was your transducer yesterday that you used for for making your own glucose sensors? Does anybody remember? You had like a flat planar electrode. What was that electrode called? Yes, it was made up of platinum. Well done, good answer. It was platinum. What format was it called? Was it in? It was called a PCB electrode, a printed circuit board electrode, right? Something that has been printed there, right? Again, based on printing technologies there. All right, so this is basically some of the things that we've already seen. And then we also saw that we were having an electronic system. You saw there was a potentiostat being used. Was it a single channel or a multi-channel? It was a single channel one, yeah? So you had 4 working electrodes, but you could only measure from 1, right? So it was a single channel electrode. And then you had an electronic system. You were getting your display. You were getting a final value of the current. You were all measuring current over time. What is that technique called? Well done, fantastic, amperometric. I think we're already having the quiz as we progress, and I can already see that you've made a lot of progress here. Well done. So you were basically using amperometric techniques. So you were having a display there, and then in the end you were plotting it and you were getting a straight line. If you got a straight line, well done, that means you got very good sensors. If you didn't get a straight line, then you go back to the drawing board and try again. That's how the whole thing works. So what I mentioned also was at the heart of your biosensor is the bioreceptor. You can have different types of bioreceptors. The main function of your bioreceptor is to provide you selectivity and specificity. I would say specificity is the most important thing. The most important thing with your bioreceptor is to provide you specificity. So you have different types of bioreceptors. You have the enzymatic biosensor, you have the immunosensors, you have the aptar sensors, and you have the molecular imprinted based sensors. And then we saw this particular nice slide on enzymatic biosensors. In the UK, when I teach this on enzymatic biosensors, this is most of the times my first, this is also what I use for questioning students as well. So I would normally say, here's how the glucose biosensor works. You've got the different generations here. You've got the first generation where you're producing hydrogen peroxide. You're using the overpotential to break it down into water and you release electrons into the external circuit. And then you have the second generation wherein you bring in a a mediator. And then you have your third generation, which is direct electron transfer. You can apply the same thing also to any other oxidase based sensor like lactate oxidase as well. You can have first generation, second generation, and third generation there as well. All right, so that's your enzymatic biosensors. And then the next example we have is that of electrochemical immunosensors. Immunosensors, as the name suggests, immuno, it always relates to antibodies, right? In this particular case, you can see you've got two sets of antibodies. This is more like a ELISA technique that you have here. You have your enzyme linked immunosensing here. You have a primary antibody which has been immobilized, and then you have a secondary antibody which comes from the top. This is more like an enzymatic, or it's more like a sandwich assay that you normally see here. So this is an example of your electrochemical immunosensor here. And then the next example we saw was that of APTA sensors. APTA sensors are basically sensors which have, what do they have as their bioreceptor? Aptamers, right? What are aptamers? Aptamers are basically strands of oligonucleotides. Well done, very well received. So you have your oligonucleotides, and then I mentioned about this very interesting theory wherein thiols can form self-assembled monolayers on gold. All of you are master's students, most of you are master's student or bachelor students or PhD students here. If you know all of this, if you understood all of this, you have actually expanded on your skill sets. It may be a bit of theory here, you've done a bit of practical there, so this is all very nice information that can go set in very nicely into your CV. And if you mention in your CV that you've done courses on wearable technology, you stand much ahead of other people, which allows you to apply for a nice PhD studentship anywhere, either in India or abroad, right? So please, as I said, first of all, I don't have to say anything more. Looking at the response, I don't have to say anything more. You've all received it very well, and you've all, actually focused and done a great job, which is a big satisfaction for me personally. All right. So this was the example of electrochemical APTA sensors here. As you see here, you've got your gold surface, you've got your thiolated aptamers, and these aptamers are self-assembled, and then as soon as you have the binding, you see a change in the electrochemical properties, and that's then measured externally, and that's normally represented here, as you can see here from these electrical impedance measurements that takes place here. Okay, let's have a look at the next one that we have. This one is something that is very, very interesting, and I say that this is the future for tomorrow. I myself am participating in conferences. I'm chairing a conference next week. I'm chairing a session on diagnostics, and there my talk is mainly on molecular imprinted polymers. We are also having this new PhD studentship coming up, so I'm meant to be talking about diagnostics from lab to bedside kind of diagnostics and those kind of things. And there my topic of presentation is molecular imprinted polymers. We've already had, Professor Khalid and myself have had a lot of talk on this particular topic, and this is going to be, I think, the future of tomorrow, right? Because by itself, it gives you a good dynamic range, and you combine this with another receptor, and you can actually increase the dynamic range. You can actually make it more ultra sensitive, right? Because what is it that you need? You need to have sensors or biosensors which are ultra sensitive, which can measure very small quantities of your biomarker. Anybody heard about circulating tumor cells? Yeah, you've heard about circulating tumor cells? Circulating tumor cells, finding circulating tumor cells in the body is like finding a needle in a haystack. That's the kind of challenge there is. And biosensing technologies, mainly molecular imprinted polymers, combined with other receptors, combined with other bioreceptors, seems to be like a technology that might be ready to accept that particular challenge, right? That's why I see a lot of promise on that, and that's why I'll be spending a little bit more time on molecular imprinted polymers to recap on the molecular imprinted polymers. As I mentioned, it's very fondly called as synthetic antibodies or plastic antibodies, and you can tailor the specificity, right? Number one, they're synthetic, they're highly stable. I gave you the example wherein we made devices with molecular imprinted polymers, sent them to Japan by courier. You can imagine they went through all the cold conditions, dry conditions, hot conditions. Japan during summer gets very hot, right? And it went and it survived. It gave us the same results that we were seeing at Liverpool, right? So you need something where you don't require a cold chain. You need something which can survive the extreme conditions. You need something that not just works in University of Liverpool, but also works here in NIT Rourkela, which also works in a lab in Kampala or in Accra, or somewhere in, where it's highly humid in Kolkata, right? Or Bombay for that matter, yeah? So you need something that works in all harsh conditions So for that, molecular imprinted polymers seem to be a very suitable candidate, and because you're synthesizing them, you're just putting a drop. Did you realize that you were just adding a drop of the solution yesterday? Did you do your electropolymerization by yourself? How much were you adding? You're adding microliters. I don't think you even went for milliliters. So if I remember very well, a day before, Rishita was preparing solutions which were about 1 milliliter, 2 milliliters. That's it. And weighing in milligrams. So you can imagine, if you can have a drop which can give you a device, you're actually looking at cost-effective solutions here. And you're making them yourselves. It's not as if you're buying them off the shelf, right? And when you buy them off, when you buy things like the enzymes off the shelf and things like that, then you're spending a lot of money. But these ones are much more cheaper. So they're very cost-effective, they're durable, and they have a long shelf life. And then I also showed you how we can make these. I showed you how we have the template molecule in the first step, which is basically your target analyte, right? So you have, let's say for example, p-tau proteins is what you're developing the sensors for your biosensors for. So you start with the p-tau proteins, and then in the next step you bring it in contact with the monomer. For example, it could be phenol red, right? So bring it with the monomer, phenol red, and then you let it interact. There would be interactions at the molecular levels. We also looked into those interactions. Do you remember that? What were those interactions? Anybody? Louder, please. Well done. Hydrogen bonds to stabilize it. What else? Electrostatic interactions. Electrostatic attraction would stabilize it. Electrostatic repulsion would destabilize it, right? So we saw all of those things there as well. And then we also looked at how then you cross-link around it. And in the whole process, when you cross-link around it, what you manage to do is you manage to maintain a particular geometry, a particular configuration, and you not only conserve the geometry, the three-dimensional structure, but you also conserve the charge around it as well. You have those charge centers there, which are ready there to kind of come and bind there. And then in the next step, what you do is you eat away the bioreceptor molecule, or you digest it, or you break it down, and then you end up having these kind of cavities that you see here, where you don't see the green molecule anymore there. All right. And I gave you the example of vancomycin, if I remember rightly, which was the example where we were developing molecular imprinted polymers for vancomycin. All right. And then after that, we also covered a little bit about dual receptors. Sometimes a single receptor does not give you the desired specificity and selectivity. Then in that case, you go and start using dual receptors. And this is another field that I would like to talk a lot about, because I've just attended recently the World Congress on Biosensors in Portugal, right? And one of the big areas which is considered to be the future, right? So this whole talk is not just about, this whole lecture is not just about the recap that we were doing, it's also identifying what is the future and where are the challenges there as well, right? So we'll be trying to do that as well. So if you talk about future, if somebody asks you, if you're sitting in an interview, what is the future of biosensing devices? How do you think ultra-sensitive devices or ultra-sensitive detection can be achieved? You can say, if a single receptor doesn't give you the desired specificity and selectivity and dynamic range, you can combine it. For example, aptamers by themselves probably will not give you the desired dynamic range. And the molecular imprinted polymers by themselves will not give you the desired selectivity, right? So in that case, you combine these two and you get the desired specificity and selectivity. And we've seen examples of that, which I'm going to recap again here. So that's how we covered the whole dual receptors, right? In terms of dual receptors, we have single bioreceptors. Khalid, can we not do it now because my Apple Watch is very sensitive to whatever you're doing there. So we have dual receptors, which can basically, wherein you combine the two receptoring systems, and we do this to improve the binding affinity, to reduce cross-reactivity, and most importantly, to enhance signal amplification. And there are several types of dual receptor systems. You can combine an antibody with an aptamer. You can combine a MIP with an aptamer. You can combine a MIP with a redox active polymer. You can combine MXenes-based scaffolds with MIPs or enzymes. All right. And then, sorry, the fonts are very small here. I gave you some examples, right? So please make sure, if you're really interested in dual receptors, that you click on these DOIs. You've got DOIs on this particular screen. You've got the slides have already been shared. The slides have already been shared. Please click on these. Have a look at these papers. You may not understand everything. Just look at what is the electrode material. You can have your own spreadsheet. And that's a very good exercise. If you really would like to work on biosensing devices and biosensors, go into your area. If you're working on environmental detection, for example, look at the environmental applications. Look at what is the electrode material there. Look at what is the bioreceptor. Look at what is the dynamic range. Look at what modifications have we done. Have your own spreadsheet, right? That will help you have a good grasp on what's going on there. So these papers will also help you in showing you how combining the different bioreceptors together gives you an enhanced sensitivity here. So you can see here going from 3 picograms to 1 picogram per milliliter. This is from 0.09 to 0.012. This one was the best example where from 9,000 you've gone to 3.3 PFU per milliliter. This is actually very, very useful here. Okay, and then you also have this one here, 63 nanometers to 47 nanometers, 1 nanogram per milliliter to 0.5 nanograms per milliliter. Now, picograms per milliliter. I mean, I've shown you examples of dynamic range where we were actually measuring in picograms per milliliter. It's not something which is imagination. You have, your batch has been very lucky that you've been exposed to some latest cutting edge technology, right? Where you've seen examples wherein we've used molecular imprinted polymers to cover a desired dynamic range for biomarkers of Alzheimer's disease, right? You've already seen that. This is an ongoing UK-Japan project. Most of these results will be coming out in papers in the future again. All right, and then I gave you the example of how MXene-based redox systems and molecular imprinted polymers can be used. This is an example wherein you have your MXenes. We saw how these MXenes are made up, and then these MXenes-based redox systems. So again, these MXenes are very useful polymers, which are two-dimensional polymers. I had these examples here. I showed you this. This is the MXene system, which is basically a two-dimensional material. It consists of a transition metal carbide here. And they're basically a class of two-dimensional materials with layered structure, which are composed of transition metal carbides, nitrides, or carbonitrides, right? This was the MXene that was presented to you. And then we saw how when we combine the MXene-based redox system and molecular imprinted polymers, we managed to get a much more wider dynamic range here. So that was the first example of seeing how you combine two different systems. Now, when I say bioreceptors, you can also have a bioreceptor combined to a nanomaterial system as well. That also could be, I mean, it's not exactly combining two bioreceptors, but it's also an example wherein you have two types of bioreceptors which are basically being combined here, okay? Two types of systems that are being combined here. One is a bioreceptor and the other one is a nanomaterial system here. And then we also looked at a second case study which was on highly porous gold this time as a nanomaterial system, and then combining them with a redox active material, which is your polyphenol red, and polypyridine, which is basically a composite here which is redox active in nature. And then I showed you some nice SCM images. If you want to see more of these kind of nice SCM images here in NIT Rourkela, go and speak to Rishita. She will be able to show you some of her SCM images of the highly porous gold. So there are facilities to do SCM here in NIT Rourkela. So if you want to look at those for curiosity as to what it happens. And then if you remember the example I gave you about these nanomaterial systems, what makes these nanomaterial systems efficient? The fact that they have, well done, they have a very high surface area to volume ratio. And how do you remember that example? By looking at the example from the body, which is that of a microvilli. So if you stretch the microvilli that you have in your intestine and keep on stretching it, and you keep on pulling it, you can reach where? To the moon. Well done, you remember the example of moon as well. I'm really glad that you're remembering all these examples. All right, so you have here is the image of this highly porous gold. So when you have a highly porous gold, you have a very high surface area to volume ratio, because within the same volume you've increased the surface area, and when you increase the surface area you increase the number of binding sites, and that's how you can increase the dynamic range as well. Okay, that's that. And then I gave you the example of how this was used for detection of p-tau proteins, and the nice thing here is we're looking at a dynamic range where you have the concentration in the region of picograms per milliliter, right? That's the thing that we need to remember. The dynamic range here is 0 to 21 picograms per milliliter, meaning that you're going from 0 to 21 picograms per milliliter. But then you need to know the sensitivity as well. Of course, the sensitivity is going to be much lower than the dynamic range, and in this particular case, the sensitivity is around 484 femtograms per milliliter. That's the kind of thing. You need to understand the different scales that we use here. You need to understand femtograms is much below picograms. Femtograms is 10 to the power of minus 15, and picograms is minus 12. Nanograms is, there you go. So all of you have a good understanding of that. So because you're going to have, when they're giving a presentation, you need to be able to identify that is this an enhancement or is this a decrease in the sensitivity, right? So that's very, very important there. So, and then finally, I gave you something which I say is hot from the press, something that has been done as recently as a few days back, some work on monitoring neurofilament light chain for Alzheimer's disease. This is some work that's going on in my lab. This is where the neurofilament light chain comes from. This is a very important biomarker of neuronal degradation. As I mentioned to all of you, neuronal degradation happens in all of us. Once we are born, neuronal degradation starts. But in the beginning, it doesn't actually happen at a much faster rate. But as we start aging, then it happens at a much faster rate, and the concentration of your neurofilament light chain increases much rapidly, right? So you need to be able to capture that, and that's also an indication of dementia, of Alzheimer's disease as well, right? And as I said, people have mentioned this in the literature, that you can predict the onset of Alzheimer's 15 years in advance using ultra-sensitive biosensing devices, right? So you are, as I said, in a fascinating field which has got fascinating applications, right? I mean, that's the whole purpose of being a scientist, right? What do you do science for? Not for prestige. You do science so that you can benefit mankind. And if you can benefit mankind in one way, even if it's a small way, that itself means that your whole objective of performing science or practicing science is achieved here. Okay, and then I gave you this example of nanomips and aptamers. The nanomips, as I said, were the system that's been practiced in my lab, wherein we are using highly porous gold as the nano system, and the molecular imprinted polymer is that of polyphenol, is that of phenol red and pyrrole, which we polymerize into polyphenol red and polypyrrole as well. And the aptamers are the ones that have been selected using a particular technique called as SELEX, SELEX. That's basically an acronym. That's how you select the aptamers, right? You can go and have a look at that if anybody's interested. People especially who want to be working in genomics need to understand about SELEX systems. So please make sure that you do some research on SELEX as well. SELEX system is used for selecting your own aptamers, and you can select your own aptamers. And these aptamers were selected by our collaborators from Tokyo University of Agriculture and Technology. And this is an example wherein they have got this pink thing. Sorry, the fonts are really small, but can you see the cursor here? So this here is your aptamer, and this is how it binds. It's got these binding sites here. This is what it uses to bind to this large molecule, which is 68 kilodaltons, is your NFL, and it binds to this NFL here as well. Okay, apta sensors for NFL, this is what I'm going to be talking about. So here, this is the results that were obtained by Tokyo University, and the nice thing here is there's one good thing and there's one bad thing, which we all saw. Honestly speaking, you made my day when you identified what was right and what was wrong here. The nice thing here is selectivity and specificity. Not the best thing here is yes, the range, the dynamic range, right? This is nanomolars. And what are we looking for? Louder. We're looking for picograms per milliliter concentration, right? That's the range we're looking for. On the other hand, so this is NFL. This is some work that has been done. This is done using nanomips here. The nice thing here is you're getting the dynamic range here. This is probably done in PBS, and then this is the next one, which has been done in plasma, and getting a limit of detection of 6.8 plus minus 30 picograms per milliliter here. And then this is when you overlay the two plots, one in plasma and one in PBS. Because PBS doesn't have any interference, right? So you can look at this. Now, what do you see here? The nice thing here is you have the desired dynamic range. What is not the nice thing here? Selectivity and specificity here. Okay, so that's what I presented this graph here. So here, look here on the left graph, which is taken from the apta sensors, has got something good. The right graph, which has been taken on nanomips, has got something good. So we would like to make the most of it. So what do? We combine these two together, and we get your apta nanomips and apta nanomip complex here, right? And then when we do the measurements, you can see here this data is only from PBS. You have managed to, so at the moment it's very hard to say what happens when it comes from plasma. I was hoping I would get some results soon, but they haven't come out yet, but I'm waiting for the plasma results. But I can tell you that for sure that the dynamic range definitely, even with the nanomips, was not giving us the actual dynamic range. We wanted to see something between 0 to 100. So here, at least one good thing that we can see in PBS is that the dynamic range has increased, right? We're getting from 0 to 100 picograms per milliliter. So to summarize, the advantages of dual systems is you can get signal amplification, you can have multi-analyte capabilities, you can improve biocompatibility, you have better reproducibility, and you have modular electro engineering potentials here as well. Okay, let's have a look at the challenges and considerations. Challenges can be how do you combine them together. So fabrication can be a challenge, it can be complex. Interfacial stability, what happens when you bring in two systems together? Are they stable at the interface? How do they react? And then data interpretation, sometimes using machine learning integrated systems, data interpretation becomes hard. And sometimes when you talk about aptamers, then you're bringing in gold as well. You're looking for gold substrates. I'm talking about thin layers, but again, it's slightly, sometimes the material cost can be high as well, right? So that was all that we learned as part of biosensing technologies. That was all that we learned as part of electrochemical biosensors. And then the message I gave you was, we can translate all our biosensing technologies, all the protocols that we develop as part of developing point of care devices, point of care biosensors towards wearable biosensors. So you can use the same. So yesterday, what did you do? You took a PCB electrode which had platinum, you functionalized it with the glucose oxidase, and you got a dose response curve, right? If you were to develop a wearable sensor, you would change the platform. Instead of a screen printed electrode, you would have what would you have for epidermal sensors, epidermal wearable ones? Planar electrodes, right? Because you're going to be looking for epidermal, what is the sample here? Sweat, right? Sweat, you don't need to break the skin, right? So what do you need? You need something more like a ring, something that can wrap around your finger, wrap around your wrist, wrap around your arm, right? So you can basically have sheets you can put a layer of planar gold on top of it, and there's ways in which you can do that. There's sputtering methods. I think you can have a chat if you want, if anybody's interested in making those kind of devices. I think the facilities are there in NIT. We have some sputtering facilities. We can get them gold coated, we can get them platinum coated, we can even do iridium oxide and things like that, so there are all those facilities here. So you can actually use this place as a base if anybody wants to pursue biosensors. If anybody wants to do a PhD, you can definitely apply to Dr. Prasann, Dr. Khalid and myself as well to see if there are any PhD positions, and we can see exactly, based on the experience, if you want to do a small project or things like that, and get you trained as well if you need that. Okay, so now we move towards wearable devices. Okay, in wearable devices, one of the things we saw that there are different types of biological sample matrices that will allow you to have wearable devices. You can have tears, you can have saliva, you can have sweat, or you can have skin interstitial fluid. Yeah, we focus mainly on epidermal and the subdermal devices. Now, just to let you know, subdermal devices, some people also call them as intradermal devices, right? For example, we call them, we as engineers, as bioengineers, call them as subdermal devices. The animal scientists, the veterinary scientists, they like to call it more as intradermal devices. It's fine as long as there's the term derma in there, wherein basically to show that you're measuring the subdermal plan. Okay, having a look at the next part, which is ISF versus sweat for biosensing. We had a look at this particular slide and we saw how sweat is easy to collect. However, the problem is it's variable and it's contamination prone. Everybody agrees with me on that? And then we looked at skin interstitial fluid. We did agree that skin is the largest organ in the body, and we have a lot more skin interstitial fluid than we have blood in our body. We have 12.5 liters of skin interstitial fluid, while we only have 5 liters of blood. All right, so that's the skin interstitial fluid. Skin interstitial fluid is preferred for subdermal microneedle sensing platforms, right? And then we also, after that, we started looking at the epidermal wearable devices. What are these epidermal wearable devices? I'll show you some examples there. We'll go over those examples one more time. The epidermal wearable devices are basically thin, flexible, conformal devices which can be worn on the skin, as you can see on the screen. They detect sweat-based biomarkers, mainly glucose, lactate, and cortisol. I think we did identify one biomarker which we thought was going to be a very useful one with with epidermal devices, with epidermal wearables. Does anybody remember that? Louder. Well done. Urea. Yeah, we saw that urea in a colorimetric manner is very, very useful to use sweat there, right? And then we also looked at how these epidermal wearables can be used for non-invasive, and they're suitable for wellness tracking as well. Okay, so we move on to the next. We move on to the next thing, wherein basically we have some examples here. We look at the epidermal wearable devices. You can see here, this is one where you use a paper strip here. You have this paper strip wherein this is used for sensing glucose from sweat, and it's combined with monitoring other vital signs, other physical sensors that you have, heart rate, blood oxygen levels, and physical activity as well. This is the epidermal wearable devices. Next thing that you have, example we have is that of a multi-layered epidermal device. Does anybody remember this? This is where you have the electronics on the top, and then you have a microfluidic network so that you can bring in your reagents, you can distribute it, and then it goes down. This is your layer which has got your reference working in and counter electrode, and then this is your adhesion layer which sticks to the body here, right? And this is where, what are these? What kind of sensors what kind of bioreceptors are these? When you see a hairpin-like structure, what does it mean? Where do you find hairpin-like structures? In which bioreceptor do you find them? RNA, well done. What are RNA-based bioreceptors called? DNA and RNA-based bioreceptors called? APTA sensors. So in the case of APTA sensors, you get a hairpin-like structure because you have complementary bases towards the end, and they can form hydrogen bonds. So they go up, and there's a loop. They have a loop-like structure there, right? That's how you get these. All right, and the round structure, the rounded part or the spherical part is what we call as the active part, and you normally have a electrochemical tag there. In this case, it's a methylene blue tag which is attached to the top of this, and then you can see this was then used for the detection of cortisol during physical exercise. That's a hormone which is a stress hormone produced during exercise. So that was another example I showed you, and then there was another example which is very useful for female health, female reproductive health. This is where you measure estradiol, which is a very important hormone, right? Why do we need to monitor hormones using continuous monitoring devices? Well done. I think next time you should be here. Excellent, guys. Well done, very well done, because hormones are released in a pulsatile manner, right? So, when we talk about continuous monitoring, when you talk about epidermal devices, or we talk about subdermal wearable devices, why do we need wearable devices? Because wearable devices are mostly required in places where you require continuous monitoring, right? There's this example of measuring glucose using a continuous glucose monitoring sensor where you get a whole profile, and there's this example of glucose being measured using a glucometer where you're getting point measurements. When you do point measurements, there's every chance that you may miss out on the low and the high excursions. You do not get any idea on the direction, the duration, and the magnitude of changes of the blood glucose concentration, right? So you need something which is more continuous. Now, as you mentioned, these are normally produced in a pulsatile manner, right? The hormones are released as pulses. The fertility is decided by the pulses. It's not decided by point measurements. Therefore, if somebody goes to a gynecologist or somebody who works in a clinic or a pathology lab and says, I'm going to take a blood measurement to give you an idea about your estradiol concentration, that's a wrong way of doing things. Maybe that's the only technology that's available. But in principle, if you want an infertility, to address the whole issue of fertility, you need to be able to measure it in a pulsatile manner. Because that pulsatile manner is actually telling you that the body is producing it in a desired way. Because this whole thing is a mix of different hormones being interplayed at the same time. While one of the hormone goes up, the other hormone goes down. That's how it normally works. Epidermal. So this is an example, a very nice example, which was published by Ye et al. In Springer Nature, and this is where they show that how they do the real-time determination of estradiol from sweat, right? Good example, but I think the problem here is you can measure it, but the problem is sweat a reliable sample matrix? I'm going to leave that up to you. If this paper has been published in Nature, clearly there's some value to it as well. And then finally coming to the example that is Mellon's favorite example of urea that you have here. You can see here how this particular, you have a variable colorimetric sweat sensor here, which is used for detection of kidney function here. And this was published by an Indian student who was in the US, wherein he's used these for multi-channel colorimetric determination of urea as well. Other examples, this is another one which is, and now after this is an example where you can start integrating the important bits here. This is where, rather than using an external battery, you're using biofuel cells, which are powered by the lactate in the body, right? These kind of things. So this is an example of the epidermal wearable devices as well. And after this, I went straight into the subdermal wearable devices, wherein I gave you examples of how skin, I think I should be having, I missed a slide, which should have been on skin. But I think if you remember, I showed you how, or I might have showed you right at the start of the epidermal devices, of how the skin the microanatomy of the skin, right? So you can tap into the skin interstitial fluid in a minimally invasive manner using microneedle arrays. And to give examples of all the translational research done, I gave you examples of some of my own work. And this was the first human pilot studies that was just done in June 2013, as you can see here. And this is where we had microneedle arrays, which were made up of SU8, and we were using an IV cannula, and we were looking at two important parameters. What are the two important parameters that you look in human pilot studies? What is the first thing that you need to ascertain? Well done, tolerability. And what is the second thing? Specificity. What else? Specificity is normally ascertained during your in vitro studies, right? When you're doing the in vitro studies, you try to get the desired dynamic range. Sorry? That it comes in tolerability. What is the next thing you look at? You look at the performance of the devices, right? So the two important things in clinical studies is tolerability and performance. How do you look at the performance of glucose sensors? Well done. Who said that? Can you say it loudly? Well, okay, Clark grid error. Well done. Clark grid error. And if you have lactate sensors, then BA plots. It stands for Bland-Altman plots. Okay, you can research all of these things, and these days, you know, life has given you things on your phone, right? Use your phone more for doing research and looking for things rather than Instagram and all those Facebook kind of things. You know, you have your whole life for the rest of the activities. This is a time when you can concentrate your knowledge and prepare yourself in race for further opportunities, right? And when the opportunity comes, grab it. There is nothing called as good luck. There's nothing called as luck. Luck is when your preparation meets the right opportunity, right? So you will go through multiple opportunities, but only when your preparation has matched the opportunity, that's when you consider to be lucky. That's when you get things in your hand. So don't rely on luck. It's all about your preparation and the opportunity. Okay, coming on to the next part, which is on tolerability of microneedles. So as I said, the first important thing we see look in is tolerability of microneedle arrays, right? So if you look at tolerability of microneedle arrays, the first thing we normally do it by two ways. One is when we use this microneedle arrays or the subdermal devices, the first thing we do is initially what we do is we look at how early do the scars disappear, right? If the scars might disappear in 2 to 3 hours, they may disappear later on. Is there any signs of inflammation? And then after that, when the whole study is completed, we then do something called as a visual assessment method. So for example, if I've done a study on Professor Prasanna here, I would at the end of the study say, okay, can you give me a visual assessment score? What score would you give to the microneedle array? What was your experience been having this for 24 hours in comparison to the implantation of the IV cannula and the comfort of wearing it compared to a different CGM sensor that has been used? So that's where you have this kind of a visual. This graph that you see here is basically a visual assessment, but that is done at the end of the study. But we also look for at the same time we also look for other things such as the scar marks disappearing as well. Okay, and then finally, the other thing that is very important that also kind of evolved during the first study was the instrumentation, right? You have to look at comfort as well, right? You're going to get a very bad score if I put Dr. Prasanna sitting here on a chair for 24 hours without giving him the opportunity to move around. He wouldn't like it, right? This was what was happening here in stage 1, wherein our clinicians who had agreed for the studies, although it was initially our phase 1 was only for 8 hours, but still, you need your natural breaks in between. It definitely is inconvenient there. And then you move from there to the bag. Now this bag was used for 24 hours. Not everybody likes to sleep with a bag, yeah? I mean, we all like to sleep as freely as possible. And then from there, something which was done in home settings, therefore we started using a PCB-based potentiostat, right? So use that for studies. Okay, so, and then these are some results, and when you plot these results on a Clark grid error, and you can see the points. So what is the main objective here? When do you say your device is working well? When points are in zones A and B, right? That's exactly when you know things are working well. But ideally, what would you like to happen? What is the best thing to happen? Everything is along this line here in the middle. Everything is in zone A, nicely controlled, because this Clark grid error is actually telling you the difference between your sensor readings versus what are the readings that you're getting from a gold standard here. Okay. That's your, that was what we did in this thing. And then I gave you examples of how we then moved from, so the first set of studies were done in steady state conditions, right? Wherein the person was, as you saw, person was sitting and relaxing, everything done in a clinical research facilities, sitting under an AC, normal temperature, nothing happening, no sweating, no movement, no artifacts during movement, nothing like that, right? The second set of studies, which were done as part of a Ministry of Defense funded project, and in that particular studies, we did the studies, we did simultaneous. Now this time there were two challenges. Number one, not just monitoring glucose, simultaneous monitoring glucose and lactate, not in steady state conditions, not in resting conditions, but in ambulatory conditions, right? This is where we did these studies and we got some good results. And what we found out is, so you could see that we were measuring glucose while the person was lecturing and also running and then we were also measuring lactate during exercise as well. And then we saw that when we take the whole thing and we bring in the AI tools here, we get much better results, which can be seen from this data here. Okay, so what you have here is basically you have your green plots. You can see there's a good correlation. We are trying to look at, as I mentioned, with your sensors, what you're measuring is the current, right? You got to then correlate it to the readings to get your predicted values. So we were using AI tools there, and we were getting good Clark grid errors for the glucose sensors, and we're getting a good Bland Altman plot for the lactate sensors, right? This was all clear to everybody? So from here, after having done human studies with our subdermal wearable devices, it was then also time to expand into other areas. And one of the areas that we wanted to expand was based on a trip that was made to a farm in Wales. And in this farm, we saw I took these pictures from this farm in Aberystwyth, and you can see here, these animals have got devices which are highly invasive. These are intraruminal devices. These devices have been fit in animals just to monitor the effects of changing their diet. And why is the diet being changed? Sorry, louder, I can't. Do you want to give the microphone here in the front? I can hear you. Yes, there's this in the front. Did you want to say? Yeah. High GI food. Why high GI food? Why are we trying to change the diet of our poor animals? Are we trying to make them fit? Do we want calorie deficit in animals so that they become fitter? Well, the problem is that the poor animals have been blamed for the greenhouse gas emissions, right? 16% of the greenhouse gases are blamed on these poor animals. So human beings, as selfish as we are, what we do, we change their diet because we have to cut down on the numbers, right? And again, this is also give an example wherein you have link between two different problems that we are talking about. One is the environmental problem of greenhouse emissions, and the other one is your biosensors. Can you see how biosensors, although is being used for monitoring of the well-being of the cows, is also in a way helping you address another problem, which is environmental emissions, right? Okay, and then when I looked at the farm, it's not as if these guys don't have point of care devices. It's not as if they don't have any biosensors at all. There are biosensors being used. You have this nice device here, these nice strips which are used for monitoring ketones in cows, right? But the problem you have there is the way you sample these animals, right? As I mentioned, the animals have, you cannot get any capillary blood from them. Can you get capillary blood from animals? Can you ask them to show their paws and can you extract any blood? No, you can't, right? So basically, we started looking into animals and we thought that the animals would be a good candidate, especially high value animals. Some animals are considered to be high value. You can have your continuous monitoring sensors, which are low cost devices, fit in a cow. You can have them fit in a horse, which is a high value. I don't know how many people have heard about horse racing. That's a big industry, right? Especially even in India, I think places close to Pune, they have this horse racing farms where they breed horses for racing, right? It's a big thing, and it's very important to maintain their well-being. It's like preparing the horses for the races. So it's very important that the animal is not under stress before you make the animal run. The animal is not going to come and tell you that I'm tired, right? You have to measure the lactate in the animal. You have to measure the cortisol in the animal. You have to measure the ketone in the animal, right? So in animals, continuous monitoring becomes much more important, right? In the animals, it's much more important to monitor their well-being, right? Okay, so the problems we have when we look at animals is number one, they have a thicker skin, right? So it's very hard to actually, so you need sturdier microneedle arrays. They have a different metabolite range, right? They have much lower physiological range. So if for us it's 4 to 8 millimoles, for them it's somewhere around 2 to 4 millimoles. So your sensors have to be more accurate in the lower side of the dynamic range, right? So that's the other thing. They have a different metabolite range. It's difficult to do reference sampling. As I mentioned, you can't get capillary blood out of the animals. The only way you can get blood out of the animals is from their tail. Yeah, you have to puncture the tail to get some venous blood. Again, if you have to do this every half an hour or every 15 minutes, would the animal like it? Exactly. And then there's also the tolerability assessment, and then there are also bioinstrumentation restraints. I showed you some pictures of some studies that were done in a farm, not in a farm, but at a place where they brought in freshly dead animals. Horrible conditions. Even the farm where you have very happy, lively animals and the calves and everything, they look all very nice. It's contaminated with the animal feces, with cow dung and everything, right? It's very hard. As I said, I gave you the example. My preferred anatomical location to do studies in milking cows would have been the udder, but the udder is so close to the floor that you would definitely have the udder contaminated. So how do you save your sensor from? That's a challenge. I will still manage to do it by whatever I have to do it, but that's a big challenge. So bioinstrumentation constraints is a big problem when it comes to animal studies. And then I showed you the design that we used. I showed you how we used these acupuncture needles, which are much more sturdier and commercially available. We used an applicator, and we used the PCB-based potentiostat here as well. And then coming to the insertion studies, these are some nice pictures to show you what happens to our needles pre and post insertion. As you can see, they're still mechanically robust. The tip still looks to be intact, no loss. Everything looks to be perfectly fine. And then finally, I showed you this nice video of this. This is a beef cow. It's not a milking cow. And then I showed you how the devices were. You can see here from here, nicely inserted. But here you can see the hair had to be shaved off, just to make sure that there is one less variable, and then you take off the applicator. Now we are designing applicators, and we're designing devices that can be inserted onto the animal without having to shave them off. Yeah. Okay. And then we also looked at the challenges and future directions. One of the biggest problems that we have and will have with these sensors, with these subdermal sensors, is the sensor drift. We have the drift problem there as well. So yesterday, if you remember, when you were doing chronoamperometry, you would have seen, or if you do a chronoamperometry with 0 millimole solution, you can see that you normally have a sensor, it drops down, it still keeps on drifting for a long time. You have to stabilize it. It has to be stabilized over a certain period of time. Normally, when you're doing these microneedle arrays, you have a much larger surface area. So when you have the sensor, you leave them to stabilize for 15 minutes till they come down all the way. Any idea, anybody? Why do we have this drift? Okay, there are two reasons for this drift. One is a normal drift, a natural drift that you would always see. The other reason is, anybody? I'll get Rishita to answer this later on, but I'll give others the chance first. So what are we polymerizing with? What are we polymerizing on the sensor surface? Phenol. Any idea now? Have you got a hint now? What can happen there? What is phenol being polymerized into? Polyphenol. So what's happening there? If the reaction doesn't complete. Louder. It forms an insulating layer. That's fine. But where is the second part of the drift coming from? Why do we need stabilization? Well done. I didn't know that you were paying so much attention on the day. So we have unreacted phenols as well, right? The unreacted phenols are also something which can cause a drift, right? Because unreacted phenols are conducting. Phenol is insulating, right? Just to make you understand the fundamentals, the molecular mechanisms, as to where the contributions can come from, right? And then the other problem we have is limited long-term data and regulatory hurdles. I showed you the example of the human studies we did as part of the DASA project. What is the big problem there? We only had ethical approvals to do studies between 8 a.m. to 5 p.m., right? Because and we were not allowed to do any studies in the night. That kills the whole objective of the studies. We get data, that's fine, and I also showed you data from one of the previous studies wherein I showed you that we know that after 24 hours, what sets in? Even before 24 hours, something sets in, which is the body's response, which is defined as the body's response. What is that called? Starts with a B. Biofouling. Well done. Yeah, so the biofouling sets in within the first 24 hours, and I showed you the results where I had those pyramids there showing you the biofouling effects. So clearly there are regulatory hurdles. There's limited long-term data. If you look at publications, this is something we've discussed so much over the last few days, is that people are publishing papers, but those papers only have got data for we've got a fantastic sensor here which can do multiplex sensing, blah, 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 but those are only done in participants over a range of 2 to 6 hours, 2 to 4 hours, and they're publishing in Nature. That's the problem we have. We don't have data wherein you can, like the animal studies that I've shown you. This is data that has been obtained over 24 hours of studies. I didn't go into much details because I thought there was going to be a lot of information in here already, and you already have seen the graphs, and you know what the important thing is to see in a Clark grid error, right? So next time when you see a Clark grid error, what are you going to be looking at? Zones A and B. And when you look at Bland-Altman plot, what are you going to look at? There's a straight line in the middle, and there are 95. How many points are between the 95% acceptance limit, right? If you've already understood how to look this, good news for you is you know how to read a paper on wearable sensors. You are now ready. So now there won't be a barrier. Earlier what happens is when we don't understand anything, we are reluctant to read that particular paper. Now you understand wearable technologies. Now you understand biosensing technologies. Now you understand point of care biosensors. Now it's not going to be a big challenge. Even for those of you who are working on physical sensors, how do you look at the data? How do you look at the performance of the devices? You now know how to measure that. You now know how to look at tolerability is going to be common, right? If the Apple Watch, before they made these Apple Watch, do you think they would have not tested these on other people? We saw the example of GlucoWatch that. And we know that the reason GlucoWatch failed was because it was not the technology that failed it, it was the adhesive that was used, which was giving rashes to people. They changed the rashes, and then suddenly they've started getting good results. And then hopefully we should have more good results based on some of the new results that have come out of NIT Rourkela as well. And the final challenge that we have with subdermal listening is getting in the AI, multiplexed sensing, real-time analytics. I did mention that when we were doing the studies in humans, the sensor was designed so that we would have a glucose sensor and a lactate sensor. However, the bioinstrumentation that was available, although it claimed to be a multi-channel potentiostat with a multiplexer, wouldn't allow us to measure more than one channel. So we had to attach two devices with two potentiostats. So imagine both the hands with two devices measuring glucose and lactate, and then asking this person to then run on a treadmill and cycle and do things like that, right? It's really hard. It's difficult. We had about 15 participants, but before the participants, I tried them on myself, and I tried to do the same thing. As scientists, you have to be conscientious, especially when you're doing the animal studies. You have to feel the pain yourself first before you propose it to be inserted in other species, whether it's animals or humans. You've got to be very, very conscientious about that. And ethics, integrity, these are all very important part of science. So the next steps we think will be bringing in the AI integration. We think we will address the challenge of multiplex sensing, and then we also think that we should be able to have something which will give real-time analytics. At the moment, all the results that we are showing you is all data that is being processed, and then we are getting the data results there. We would like something to be similar to the CGM sensors that we have. Even the CGM sensors, it's processing over time, and then it's throwing a data value every 15 minutes. We would like to have something which is a bit more faster. An electronic architecture, which I think hopefully will be designed with Professor Khalid's group, wherein we have real-time values. Oh, your value is this much. But then there are pros and cons there, right? As I said to you, the reason the continuous glucose monitoring sensors had poor acceptance at the start was because of the fact that they are devices that are implanted deep into the body, right? When your technology is fighting the body is definitely going to win, right? So how do you address the problem of biofouling? You can address the problem of biofouling, number one, by bringing in a membrane that is perm selective, that is only selective to the glucose molecules and does not allow the proteins that are being produced by the body to cover the sensor surface. That's number one, right? Over time, it will go and cover the membrane, but none of them will reach the sensor surface. And what will happen, even if it covers the membrane, the amount of glucose that will diffuse through the membrane onto the sensor surface will go down. So there's no way you can fight it, right? The body is definitely going to win. The other thing you can do is you can do a proper study using machine learning tools or deep learning tools, bring in your AI models and then predict values. But again, those values would be predicted values. Those will not be the absolute accurate values. You will always see a difference in your blood glucose reading done between two devices in the body because they use two different AI models, right? I think that was all about the presentation I had for you. That was all that was presented. What I'll do is I'll give you 5 minutes and I'll open the floor for any questions. This is my email address. Some of you have already sent me LinkedIn requests to join to this thing. I have already approved some of them I haven't. I just want to be sure because I keep on getting a lot of requests, especially since I've been to the World Congress on Biosensors as well. I just want to make sure that it is relevant to your interest as well. And I'll be more than happy to connect via LinkedIn, and I'll be more than happy to share my research insights. Most of it normally comes up on LinkedIn every time we have a presentation at the conference or we have a paper publication. So if you are already attached to or connected on the LinkedIn site, that's a good thing. I'm not on Instagram. I don't do Instagram. That's for you young generation. I'm too old for Instagram. Facebook is more personal, so I don't give my Facebook to anybody. But yeah, I think LinkedIn is a good site, a good platform to connect and to keep in touch.
Hello. Am I audible? Yeah.
Yeah. You're
so, Sanjeev, just one of in the slides you talked about the MIP based sensors.
Yes.
So, the fabrication processes you mentioned, there are certain challenges as well.
Yeah.
So, do you think that currently the state of art says that this MIP based sensors can be a scalable way of making those sensors which anybody can use it, or still there is a lot of the hurdles which need to be overcome? What's your opinion?
Well, the slides that I showed you, I mean, for the lab scale, for the research purposes, that's perfect because you're talking about electropolymerization. Now, it's a good question you've asked. All right. This is a nice question Dr. Prasanna has asked, and I want the answer to come from the audience, right? When you talk about medical devices, what is the most important thing that you need? Most important thing to make it acceptable globally?
Standardizations.
Standardization. Before that, even standardization. Sorry? Biocompatibility, what else? What is the most important thing when you talk about medical devices? When is a device most easily acceptable? Louder.
Safe.
Cost. So to make it cost effective, what do you need for making it cost effective? Well done. Sustainable material. What else? How do you make it out of? So what do you need for that to make them for manufacturing? Scalable technologies. Right? So if you have something that is, so at the moment in the lab, we are doing electropolymerization. The question to you is, and then that's where you're going to answer Dr. Prasanna, is electropolymerization a scalable technology? I want you to think about it. Is it or is it not? I think it's not. Why? Because imagine, how many engineers do we have in the room? How many engineers? All right, engineers, come on, help, come and save me. When you talk about, if you were to make electropolymerized, if you were to have molecular imprinted polymers, I mean, basically you need polymerization. Is electropolymerization the way forward when it comes to scalable technologies, or would you prefer something else? Have you heard about robotic technology, robotic arm, automatic dispensing? You need something like that. Can you do polymerization? Can you do electropolymerization with a robotic arm? You can, but it's going to be very time consuming. It's going to be very laborious. So you need something where you do the polymerization without applying an electric field. That's your answer.
Okay, one more question. So in the examples which you showed, phenols were used for polymerization and making the polyphenols. So there could be the other type of the also materials which could be used.
There's a lot more. I mean, there is actually a review article on molecular imprinted polymers, which I think everybody should read. There are many of these molecular imprinted polymers because there is a list of materials.
Okay.
And you can, as I said, excuse me, there is a list of materials and you can actually use those materials. You can combine those materials. You can combine the good things of different materials. Something would be better conductive, something would be better redox properties. Something will work at a different potential, something will work at a much lower potential. There's a lot more different materials you can combine, and there's a list, organic compounds. I mean, any chemical engineers in the room? Can you give me a rough idea as to how many organic compounds you have? A lot, and a lot of phenolic compounds, a lot of these, the rings which have aromatic ring compounds or cyclic ring compounds, a lot of them have got redox properties, and you can exploit them. But I like the idea of sustainable materials. Well done.
Any other question, guys? So the floor is open for questioning.
Are we all ready for the quiz then?
I think they are ready for quiz.
All right. If there's no more questions, I mean, looking at your responses, me personally, I'm mightily pleased. Very, very pleased. Thank you so much, all of you, for paying attention, attending the lectures regularly, and answering. As I say, my students inspire me to. I mean, it's everywhere, whether it's in the UK or whether it's in India. India even so a lot more. It inspires me, to kind of give my best. I cannot explain to you. I'm not in the best of. I've overdosed myself with paracetamol and cetirizine, and to stand here so that I can give you a final talk. But I'm really, really happy that that all of you have been very responsive. Thank you so much for that.
Thank you, Professor Sanjeev.